In this session, let us study the mode of action of chymotrypsin. Now, for example, this is the enzyme chymotrypsin and this is the substrate. Substrate means the protein which we have taken from our diet, through our diet. So this one is the substrate and this is the enzyme with three amino acids, that is catalytic triad, aspartate at position 102, histidine at position 57 and serine at its position 195. As we have already discussed that normally the hydroxyl group of serine residue, this particular group of serine residue is not acidic, and this is true for all other serines of chymotrypsin. Only serine 195 means this serine becomes acidic due to this unique constellation of three amino acid residues because of the specific protein folding, that unique folding of the protein. Now, what is the importance of this acidic serine residue? Let us discuss about this serine. See, this oxygen anion of serine is negatively charged. It is able to make a nucleophilic attack on the carbonyl carbon. This is the carbonyl carbon. So this oxygen anion makes a nucleophilic attack on this carbonyl carbon of the peptide bond of its substrate. This is the substrate. This is the peptide bond of carbonyl carbon. So this oxygen makes the nucleophilic attack on this bond of carbonyl carbon and a short lined intermediate is formed. After that, this bond gets loosens. Now after loosening, then a water molecule can easily hydrolyze this particular bond of a substrate, and this is the specific site, active site of chymotrypsin where this substrate is present. This is the active site of chymotrypsin where this substrate is present. This is the active site and it allows only bulky aromatic amino acids to bind. These are bulky aromatic hydrophobic amino acids. As we all know that this enzyme chymotrypsin is specific to aromatic residues only. So at the active site only bulky aromatic amino acids can bind. And after this binding, this is the susceptible peptide bond which breaks during which remains close to this serine residue, and after that this bond gets break and this whole peptide, polypeptide, the protein which we have taken from our diet, it gets break into smaller peptides. Now I want to tell you that basically what happened in chymotrypsinogen, as this is the active site of chymotrypsin. This is the active enzyme chymotrypsin, but when it is in the form of chymotrypsinogen, this substrate binding site means this active site is blocked in that case, and hence the enzyme is inactive. So when this particular substrate binding site is blocked, this enzyme remains inactive, and this is in situ activation. First of all, trypsin gets activated. Trypsin enzyme gets activated, and after its activation, it helps in activation of chymotrypsinogen to chymotrypsin. Means chymotrypsinogen gets converted into chymotrypsin with the help of this trypsin enzyme. And in chymotrypsin, this active site gets exposed. That's why this is known as active enzyme. When this active site gets exposed, so this enzyme becomes active.